Hello and welcome to this video presentation on our work. I'm Dr. Aidan Mead, and this work is an inter-comparison of the performance of deep learning and traditional machine learning for classification of cancer and pre-cancer stage with hyperspectral imaging applied to tissue biopsies. The diagnosis of pre-cancer stage remains a significant challenge to pathologists. Here we used FTIR images of esophageal tissues, which were required for the purposes of classifying inflammatory and precancer stage in esophageal cancer. A total of 191 samples from 149 patients, which were mounted as a TMA, were imaged with a Perkin-Elmer Spotlight 400 FTIR microscope, where tissues were divided diagnostically into 5 classes. A parallel slice to that used for FTIR was stained with hematoxylin and eosin for histological verification. Using the FTIR images, we tested our algorithms in classifying images according to histological class in a 5-class and 3-class challenge as shown. Our classification pipeline then involved several stages. Firstly, was a background removal stage, which removed non-tissue relevant spectra from around the image. Then tissues were scatter corrected, and the first diagnostic stage involved segmentation using an automated k-means approach, which is referenced below and identifies the optimal number of individual spectral clusters within the image. Subsequently, cluster spectra were classified using partial least squares discriminant analysis. In the second approach, images were classified after scatter correction using a deep learning approach. All algorithms were trained, validated and tested with data split randomly by patient in a 60-20-20 split. We used several freely available architectures for deep learning as listed here in the box, and we depict a sample structure for LINET5 which we modified as shown to allow input of hyperspectral imaging data. The automated k-means approach successfully produced false colour images which matched well the H and E stained section. Though when we subdivided spectra by cluster, certain clusters did not have sufficient data for training and testing and were excluded from further analysis. In general, it was found that a discriminant analysis approach performed relatively poorly at the 5-class task, though certain substructures performed relatively well at identifying precancer in a 3-class task, as shown here. Overall, this algorithm performed excellently in a 2-class normal versus cancer task as shown by the receiver operating characteristic in blue. Though again, the classification of precancer stage remains a challenge, particularly so for classification of esophagitis from normal tissue. All deep learning architectures were found to perform similarly poorly in respect of the 5-class task, but certain architectures performed reasonably well at the 3-class task, with the 2-class problem not tested here. In conclusion, it was found that both machine learning and deep learning techniques performed reasonably well at classifying pre-cancerous stage from cancer. Deep learning does offer an advantage over machine learning in that image segmentation and pre-treatment stages can be eliminated, which can introduce efficiencies into the diagnostic workflow. Ultimately, this study used a small sample set, and a broader sample distribution of images by class is required to effectively train and test algorithms in challenges such as this. I'd just like to acknowledge my colleagues in Technological University Dublin, in Trinity College Dublin for supply of tissue and diagnostic information, and the Health Research Board in Ireland for funding. Thank you for your attention.